Hi everybody. So this video is meant to help you through the enzyme worksheet activity we're doing in class today. And if this is in case you couldn't make it for the Google Meet because we did this together. So we're starting off by talking about enzymes here and explaining what they are. So I want you, as we go through this, I want you to try and figure out the answers before I give them to you on here. I don't want you just filling them in, because at some point you're going to have to know this on your own. So you can pause yourself before I release the answer, and then maybe that'll help you quiz yourself. So think about it for a second. What are enzymes? They are a type of protein that helps reactions occur inside your cells. So they're going to help speed up all of the chemical processes that occur inside your cells. If we did not have these proteins, these enzymes, nothing would happen and we would be dead. I mean, nothing would work in our bodies. Even for plants and all organisms, they all have enzymes. An enzyme is a biological catalyst because, the term catalyst is any substance that makes a reaction occur. And a catalyst could be something in a beaker in the chemistry lab that helps a reaction occur, like gasoline for a fire, that's a catalyst. But the term biological catalyst is what's making this relatable to living things. So an enzyme is a biological catalyst because they help reactions occur inside living things, okay? And for enzymes in general, some of them are a little bit different, but in general, you can recognize them because they end in ASE. So we made it nice and big to help you remember. And in these boxes here, we're going to put in 3 examples. So we're going to use lipase. Lipase is named to break down lipids in our bodies, fats, in our digestive system. We've got amylase, that's another enzyme in our digestive system that will help break down proteins. And then polymerase, remember a polymer is a large compound, so polymerase is actually something that's going to synthesize or make bigger compounds like DNA polymerase. Okay, so now we're going to identify what the different parts of this reaction are. And for a chemical reaction, I'm just going to delete myself here. For a chemical reaction, whenever you see these arrows, it's showing you the direction of the reaction. It's showing you a process that's occurring. So we're starting here on the left, and we're starting with these AB shapes, and these are our reactants, and then this red shape. So for the AB shape, we know those are the substrates because you can see they get changed before the end of this reaction. The red thing is our enzyme because it will not get changed. It does not get used up. It doesn't change. It's still there at the end. When they're stuck together, we call that the enzyme substrate complex. And then just an added note, right here, this cutout area of the enzyme, that's called the active site. And I didn't label it on here. You could label it so you remember. And then, of course, here at the end again, we still have the enzyme. All right, so going on to our next diagram, this is what it looks like for you. We have 2 types of enzyme reactions. I want you to take these pictures over here and click and drag them to show what kind of reaction they are. Remember, digestion is breaking things down. Remember that when you eat your food, you digest it, you're breaking it down so that those particles are small enough to get into the cells of your body so you can use those particles. And synthesis, a synthesis reaction is when you're making something out of smaller things, usually something bigger. So let's take this first one, and we're looking to see, usually we'll just assume they go from left to right, these reactions. So we have these 2 circles going into this enzyme. There's our enzyme substrate complex, and then we've got different circles. They're connected now, so they're bigger. So it looks like we're making something, so that's going to go in our synthesis reaction. And then this next one here, we're taking a purple thing and a green thing, and when I look through quick, the green thing doesn't change, so I know that's my enzyme. So I'm going to watch to see what happens with this purple thing, and it looks like it gets broken into 2 pieces instead of one big one. So to me, that sounds like digestion because we're breaking it down. And then we have this reaction. We've got the blue thing, you can see that stays the same, so we know that's our enzyme. So the tan piece is our substrate, and it looks like these pieces are stuck together. And then we've got two smaller pieces at the end. So to me, that looks like digestion as well. Let's take a look at this one. All right, so we've got a triangle and a circle fitting into this piece. We've got two pieces, and they become joined by the end. So that looks like a synthesis reaction because we're making something bigger. And then this one, we've got piece A. This greenish piece stays the same throughout, so that's our enzyme. Piece A ends up splitting into two, so that's digestion. And so that takes care of all our pieces. All right, last slide. Just to kind of review this information, what type of organic compound are enzymes? We named that in the beginning, but if you remember, they're either going to be a protein, a lipid, a carbohydrate, or a nucleic acid, and they belong in the protein group. And what determines how an enzyme functions? I had you add that to your notes last Friday, and it's the shape that determines their function. You can see how this cutout region of this enzyme will only fit this particular substrate because of its shape. So there are factors that can denature enzymes or change that shape of the active site so they don't work anymore. And those two factors are going to be pH and temperature. If you think about it, when you cook an egg gets cooked by temperature. An egg is mostly protein, right? And enzymes are types of proteins. You can't change it back. So it's been completely denatured and changed permanently. Okay, so now we're going to label this diagram with these terms: substrate, enzyme, the enzyme substrate complex, our product, and our active site. So the first one, our substrate, is the piece that we're starting with that becomes different by the end. So there's our substrate. The enzyme is the thing that doesn't change, so that's going to be our gray thing, okay? The enzyme substrate complex, typically in the middle when they're all joined together. And then our product is what it's already kind of labeled for you, your product's there. And then the active site, it's the outline of that cutout region right in the enzyme. And then explain why substance X right here, which is our enzyme, would not be likely to digest a different organic compound. If you had something that had a different shape to it, it's not going to fit in the active site. So substance X has a specific shape and can only fit with one type, it should say, of organic compound. Okay? Hopefully this made all sense to you. If not, go through it again. And if you have any other questions, let me know.